recorded.
So thank you, Dr. Xie, for giving us a talk. Dr. Xie did his bachelor's and master's both in China, followed by a Ph.D. at the University of Texas at Austin, followed by his postdoctoral work at the University of North Carolina at Chapel Hill. And now he's currently an associate professor at the Ohio State University, doing a lot of research on hearing loss and cochlear nucleus. And we're really looking forward to your talk.
Thank you for the introduction. Hi, good morning. I know it's very early there. So I would like to talk about the research findings from my lab in the past several years, especially about the mechanisms of age-related hearing loss in the cochlear nucleus. So first of all, I don't have any conflict of interest to declare. The outline of my talk is that I will briefly give an overview about the auditory system and the general plan for sound processing in the system. And then I will talk about the changes in the cochlea that we found recently in the past 10 years or so, about changes in spiral ganglion neurons, different subtypes, and pathological changes during hearing loss. And then I will focus in the cochlear nucleus, especially about the connection between spiral ganglion neurons and the neurons in the cochlear nucleus. So that includes the autonomic nerve synapse and the postsynaptic cochlear nucleus neurons, and what changes it goes through during aging. And then how that might underlie the mechanisms of age-related hearing loss. And finally, I'll give a summary. So first of all, here are the diagram of the auditory system, which we know that on the left is the periphery auditory system, on the right is the central auditory system. Let me see if I can show the laser pointer. Oh, sorry. So, okay, sorry. I'm not sure you can see my mouse. So in the periphery system, we know that this is where the mechanical sound vibration is transduced into electric signals, basically by the hair cells. And then the signals are passed to the spiral ganglion neurons, which form the auditory nerve, and then goes into the central nervous system. That the first site is the cochlear nucleus, which is the focus of my research. And then signals are redistributed to different parallel pathways in the brainstem, and then goes all the way to the inferior colliculus and to the thalamus, and eventually end up in the primary auditory cortex. This is where we form perception of the sound. So how sound is processed in the system. And I would like to start with two basic properties of sound, which is our frequency and amplitude. As shown in the wave here, in red, we know frequency, they're low frequency, high frequency. It's the rate where the pattern of alternation is repeated. So on top is lower frequency, which correspond to low tones. And then bottom, they're high frequency or high tones. And we perceive as pitch. And then for amplitude, which is shown in blue, that the waves have different amplitude. Smaller amplitude correspond to lower levels of sound. And then louder amplitude correspond to bigger amplitude. So these two basic properties actually can be used to describe any complex sound. So in other words, complex sound is mixed waves of different frequency and amplitudes. And like music or speech, we can break down based on these two basic basic properties. And the challenge for the auditory system is that how to process a complex sound and to produce perception in our brain. And then the master plan of the sound processing is that in the periphery, our inner ear or cochlea will break the complex sound by these two basic properties, by frequency and by amplitude, and then pass information through parallel inputs into the central system. And then in the central, we'll reorganize and mix match all this information, and then do all kinds of neural computation to extract useful information, and then filter out noise and unrelated signals. And then eventually we form perception. So again, these two basic properties are key throughout the system. Sorry. Okay, first of all, frequency, we know that in the cochlea, we have the basilar membrane that provide the physical separation of frequency, give us the tone-topic map. Basically, at the base, there's the basilar membrane is more sensitive to the high frequency sound, and then at the apex, it's low frequency. You can consider this as a it's like a piano keyboard that lines from lower to higher, or from higher to lower. You see from the base to the apex. And then complex sound is broken down by frequency along this basilar membrane. And this kind of a tone-topic map or tone-topic organization is maintained throughout the whole auditory system, from the cochlea to the cochlear nucleus, nuclei in the brain system, and all the way to the cortex, and even beyond the primary cortex. So this is how frequency is encoded. And at the basilar membrane, we have roughly 3,500 inner hair cells along this tone-topic map. So we can consider every inner hair cell as an individual frequency channel that carry this information about that specific frequency. So this is frequency encoding. And intensity is that, here's a diagram show that there's one row of inner hair cells and 3 rows of outer hair cells along the basilar membrane. And the inner hair cells are the ones that carry the most sound signal to the central auditory nervous system. So every inner hair cell is actually connected to 10 to 20 different spiral ganglion neurons. And this spiral ganglion neurons, they're not identical. They have different subtypes. And they carry different aspects of sound information from that frequency or from that inner hair cell. So this spiral ganglion neurons, they are sensitive to different intensities, and they have different dynamic ranges and have different rate coding. So every inner hair cell, I mean, they have different spiral ganglion cells that encode different sound intensity. That's how the intensity is encoded from the cochlea. And again, this information, it goes in parallel and processed, is sent through auditory nerve to the cochlear nucleus. And you can see to the right that every channel can consider that there's a bunch of different cell types receive information from the same channel, and then process all the information regarding about this this particular frequency band. And then as information is further processed in parallel pathways along the auditory system, and then before we form a perception. So that's how, I mean, the general plan for the auditory system to process frequency and process intensity. And by doing this, we can actually perceive any complex sound will be represented in our brain. So in the past 10 to 15 years, no, it's longer than that. So I would like to talk about specifically different subtypes of spiral ganglion cells, or type 1 spiral ganglion cells. These are the ones that connect with the inner hair cells. I mean, type 2 spiral ganglion cells connect to outer hair cells, and they only like 5% of them. So I will not talk about that in this talk. So type 1 spiral ganglion cells, back in the late 70s and early 80s, Charles Lippmann did the pioneer work that by recording from single ultra nerve fibers, what he found was that this ultra nerve fibers, they have different response properties to sound. So if you play tone bursts at different sound intensity, and what he found was that there are basically 3 different subgroups for the type 1 spiral ganglion cells. There's one group, it responds well to low-level sound, I mean, to all levels of sound beyond a certain threshold. And even without sound, this ultra nerve, they fire spontaneous spikes at a higher rate. So he called them high spontaneous rate and low threshold ultra nerve fibers, or spiral ganglion cells. And then there's another subgroup. They only respond to higher intensities, and they do not respond to medium or low intensity sound. And the spontaneous spikes is very low in firing rate. So he called them low spontaneous rate and high threshold fibers. And then, of course, there's a subgroup in the middle. It's called medium spontaneous rate and medium threshold fibers. And then anatomically, what he found was that the high spontaneous rate and low spontaneous rate fibers, they locate at different sites of the inner hair cells, which is it's a very elegant organization. And we know that by combining all these different subgroups, subtypes of spiral ganglion neurons, we'll get a full representation of all the intensities at this specific frequency band, which we can use for complex sound. And then more recently, what he found was that it's the type 1Cs, or low spontaneous rate fibers, that they're more sensitive or more vulnerable to damage, especially to noise, and also during aging, that they're preferentially damaged during hearing loss. And the consequence was that now you lose the sensitivity in responding to high intensity sounds, even though you can have normal hearing threshold. People develop problem is called like speech detection in noisy environment. When you have high background noise, you saturate the type 1A or maybe type 1B spiral ganglion cells. And when you have a louder signal without type 1Cs, you're going to have problem in detecting the signal in noisy background. Yeah, that was thought to be the mechanism underlying hidden hearing loss. So anyway, the problem is that we know all this happens in the cochlea. So what about in the central auditory nervous system? And does this change in the cochlea correspond to specific changes in the central auditory system? And what are the underlying mechanisms? And then one key site is studying this is in the cochlear nucleus, where the cochlear nucleus neurons receive all the input from spiral ganglion cells. So as the diagram show here, that yeah, all different subtypes of type 1 spiral ganglion neurons give input to cochlear nucleus neurons, and then the connection of this central auditory nerve synapse versus postsynaptic cochlear nucleus neurons, this connection, and also the postsynaptic responses, how do they change during hearing loss was unknown. So that's how I got interested in studying this. And I was I'm a trained electrophysiologist. So I want to know what are the physiological changes during age-related hearing loss, and what are the morphological changes to the synapse and also to the postsynaptic bushy cochlear nucleus neurons. So again, in the 80s and 90s, this question was already, studied by doing the single unit recording, the same technique Charles Lippmann used, that basically use a very sharp electrode, record a single ultra nerve fiber in live animals, mostly cats. And then, I mean, physiologically, you can define the subtype based on the properties like different threshold, different dynamic range, tuning curve. And then here's a summary of all the fibers. You get a audiogram of the animal hearing, basically. And then after the recording, a dye is injected into the ultra nerve. And then over time, the dye will diffuse through the nerve and gets into the cochlear nucleus. And that's where they sacrifice the animal, and then they do the fix and the dye tracing. And then they can figure out where the ultra nerve goes in the cochlear nucleus. So here's a overall drawing of the cochlear nucleus slice. You can see this fiber goes in and bifurcates. There's one branch goes to the ventral cochlear nucleus. The other branch goes through another part is called posterior ventral cochlear nucleus, and then end up in the dorsal cochlear nucleus. And here's a magnified view that you can see the fibers. There's branching out, and then you can see that it goes within the cochlear nucleus and end up with synaptic terminals. They're terminals that get in touch with the neuron. And our high magnification, what they found was that, okay, some of the synapses are very big. It's called end bulb of Held synapse. They target the cell body of the postsynaptic cochlear nucleus neurons. Here's the morphology of the synaptic terminal. And then based on the physiological recording, they can classify this as a low spontaneous rate fiber or synapse, and on the right are high spontaneous rate synapse. And they can study the morphology of the synaptic terminal. However, I mean, this technique is very tedious. It's very low yield, and they have no, they know nothing about the postsynaptic cell, because, I mean, to do this, you have to sacrifice the animal, fix the tissue. It's really hard to get all the information pre and postsynaptic in terms of physiological properties. So, and then there, 6 or 7 years ago, there are 2 papers were published using the technique is called single cell RNA sequencing. And what they found was that there are different molecular markers that can separate different subtypes. Particularly, there are calretinin, calbindin, and PAL4F1. There's 3 unique molecular markers can separate these 3 different subtypes of type 1 spiral ganglion cells. And then after this, we got very interested, because now we have a tool that we don't have to do in vivo recording. And with the molecular marker, we can identify different subtypes. So that's how we started with this project, that using slice recording to study the properties of different subtypes. So here's the experimental approach that we're using CBA-CAJ mice of different ages. So we have different age groups. The normal hearing are 2 to 5 months. They have very good hearing. And then we use really old mice, like about 26 to 30 months of age. They develop age-related hearing loss. And then we want to study their circuit and how that changes in terms of morphology and physiology at the connection site between the spiral ganglion cells and the postsynaptic, it's called bushy cells. Again, we get the mice, we test ABR to evaluate hearing, and then we sacrifice the mice. We cut the acute brain slice that contains the cochlear nucleus here. So you can see the auditory nerve goes into the cochlear nucleus, and then there's the two branches. One branch goes to the AVCN. There's another branch goes to PVCN and DCN, just like what I showed in the previous slide. And then we under the high magnification of the microscope, we can actually see the neurons in the AVCN, or I mean, anywhere you look, you see neurons. And we're focused in the anterior ventral cochlear nucleus. There's a special cell type. It's called bushy cells. They have a unique function about processing the temporal information of the sound, which are kind of our model to investigate the question. So again, I mean, I'm showing you the young mice and old mice. In young mice, you can see nice-looking cells. The cell body, I mean, very nice looking. It's actually, the actual image is better than this. And then in old mice, we also can find the neurons, except that inside the neuron, we see this junk accumulation. It's called light fusion because cells, they have waste proteins. I mean, at young age, the waste proteins are always effectively digested and cleared off. But during aging, the efficiency decreases and waste proteins kind of accumulate inside the cell and become junk. And then we can see that under the microscope as, I mean, in almost every neuron we see this. But regardless, we can still record from the cells using the technique. It's called whole cell patch clamp recording. So we found the cell, we use a very sharp electrode, advance against the membrane, and then we use a suction to break the membrane to get access to the inside of the cell. And then this is the example that we fill the cell with a fluorescent dye, and then you can see the morphology, that this is cell body, and there's a dendrite, and this is axon. And what you can't see are the ultranerve synapses that are connecting on the soma and also on the dendrite of these bushy cells. So anyway, after we get access to the cell, and we can record physiological activities by either injecting current through the recording electrode to the cell to describe the intrinsic property of the cell. How do they fire? What kind of channel they have? By doing this current injection. And alternatively, we can give an electric shock to the ultra nerve, and then to activate the nerve and trigger activities at the synaptic terminal, which induce the synaptic transmission, and then pass the signal to the postsynaptic bushy cell. And then to evaluate the function of the synapse. And this is one of the recordings that we give a brief shock, and then we recorded action potential from the postsynaptic bushy cell. And this is called current clamp recording. And in a different mode, the voltage clamp recording, we can block all the intrinsic activities of bushy cells and only record the signals from the synapse. Which I'm not having time to show you today, but basically we can evaluate the physiological function of the synapse and also the bushy cell readout upon the ultranerve input. So to the right, I mean, after the recording, we fill the cell with fluorescent dye, and then we fix the tissue. So we remove the electrode. The cell kind of reseals and traps the fluorescent dye inside. And then we take the brain slice, we fix, and we do immunohistochemistry against different molecular markers. So for example, vesicular glutamate transporter 1 labels the vesicles inside the synaptic terminal. So basically, it's a synaptic marker to label ultranerve synapses. And then this is another marker, calretinin, we know that it labels type 1A spiral ganglion cell. So it not only labels the synaptic terminal, but also the connected axon or auditory nerve. If you show, see a diagram here, E1, it will label the terminal and the auditory nerve fiber. And by merging the different stainings, we can see that here are postsynaptic bushy cell that we recorded, and here's one of the auditory nerve synapse. It's a type 1A subtype. You can see the axon, and then it wraps around the cell body, and then it contains the vesicle that inside the synaptic terminal. And then we take an image stack to cover the whole structure of the cell and the synapse, and then we do a 3D reconstruction to get the structure of both the synapse and the cell. And we made a video. I will show you how we made it. So here's the image that you can see the red fibers, auditory nerve fibers, and then the magenta filled cell that we recorded. And then the greens are the VGLUT1 staining of the synaptic terminals or glutamate vesicles. So I will show you here. So we render surface of the vesicles and the fibers. Now we focus on the cell, and we focus on the vesicles inside the synaptic terminal. So what you see here are two different types of vesicles. So the yellow is actually the overlap of green with red. It shows the synaptic vesicle inside the type 1A synapse, which is labeled in red. The green only terminals are non-type 1A synapses because they do not express calretinin, and it's not showing any other markers. So it just shows the labeling of the vesicle alone. So by doing this staining, we know that this cell receives at least two different auditory nerve synapses. There's a type 1A input, which is shown in yellow, and then there's at least one input from a non-type 1A spiral ganglion cells. And then we repeated this study in many cells. Basically, we want to correlate the morphology of this synaptic input with the response property of the bushy cells. So, and then here are 4 different examples. So this is one of the bushy cell. It receives majority of this type 1A auditory nerve inputs or spiral ganglion cell input. We quantify by doing the volume measurement. It's like 88% of the vesicle volume are from type 1As, and 12% are from non-type 1As. And the response of this bushy cell, I will skip the intrinsic properties. I will go strictly to the auditory nerve stimulation. And this is a long train of electric shock at the auditory nerve at 100 Hertz. And the response of the cell is by firing tonic action potentials throughout the duration of the train. And then at the end, it will show that some spikes, even though there's some failures, but you can see most stimuli trigger single action potentials. And then here's another example that this neuron receives majority of non-type 1A inputs. So the type 1A volume only has like 2%. A majority of other inputs are not from type 1A spiral ganglion cells. And then if we give the same stimulation, and the cell, very interestingly, it fires only transiently at the beginning of the stimulus train. And then it stops firing. So, and between these two types, there are intermediate levels. Some cells that receive like 59% of type 1A input, it fire in this more adaptive pattern that it fire lots of spikes at the beginning and then gradually slows down. And then there are cells that have 35% of type 1A input. It fire more spikes at the beginning and then stops. So we saw this different patterns of spike that in response to the outer nerve stimulation that correlate with how much type 1A input they receive. And here's the summary to show the findings that we found different type of bushy cells, they receive different component of type 1A versus non-type 1A input, and they fire different spikes. For this one, they fire tonic spikes, and they have majority of type 1A subtype input. And for this one, for majority of type 1C or mixed type 1B1C input, and they fire only transiently to the stimulus trend. So now there's a correlation that you have frequency and you have intensity separation in the cochlea, and the information in the cochlear nucleus is kind of mixed and matched in different bushy cells, and then which correlate with very distinct response properties. So that's under the normal hearing condition. Now, what happens during age-related hearing loss? We know age-related hearing loss is the prevalence increase greatly in humans beyond 60. And the impact of hearing loss affects both frequency and intensity. Frequency, we know that we have high frequency lows first, and then gradually develop into low frequency hearing loss. And the intensity, we know in the cochlea that type 1C spiral ganglion cells are more preferentially lost before we develop more severe hearing loss. And what happens in the cochlear nucleus at the connection between spiral ganglion cells and bushy cells? So again, we did this in mice. I showed you what happens in normal hearing young mice, and then we repeated the same study in the middle-aged and really old mice. So here are the ABRs from these three age groups. You can see that the hearing threshold elevate with age, and then the wave one amplitude significantly reduce over time. And especially in the old group, the wave amplitude are only like 10–15% of the young group. And then hearing threshold is 75 to 80 decibel. This is to clicks. And then I won't show you the detailed experiments. This is a summary slide to show what happens to the changes in that synaptic terminal. So at young age, we see nice looking VGRU21 stainings of different subtypes in yellow and green. And in middle age, we see the total volume kind of decrease. And in old mice, the synapse gets shrink even more and volume decrease. And especially the green, which are type 1B1Cs, decreased even more by percentage. Here's a summary that we see the yellow subtype over time, it decreased by 30% or 30 or 40%. And relatively, the green decreased like 60%. So there's a more preferential damage of the type 1B1C synapses. And postsynaptic cells, based on the dominance or prevalence of the type 1A synapses, the percentage of the cells also decrease with time. So the idea is that you have the different subtypes of spiral ganglion cells. I mean, all the terminals degrade with time. However, there's more preferential damage to type 1C synapses in the cochlea, and also same thing happens in the cochlear nucleus in the central synapse site. And then we examined the postsynaptic bushy cells. First of all, the morphology of the cells. We reconstructed Bushy cells from the young group. You can see they are all like very nice looking, showing the cell body, axons, and very heavily branched dendrites. In the old group, what we saw is that some of the cells, you have relatively normal dendrites. However, lots of Bushy cells in the old group, we saw that the cell body are shrink a lot, and then the dendrites, they have a heavy loss of dendritic branches, and the branch length is also greatly reduced. So there's dendritic degeneration, the shrink of the cell body size, kind of morphologically changes with age. And we found that this group of like more damaged Bushy cells, they tend to receive more type 1B1C synapses, kind of corresponding to the more damage of the type 1C sphere ganglia cells, or reduced inputs from those cells. And then we studied the more intrinsic properties about the Bushy cells. We just published a paper last year that we investigated the different channels, different potassium channels. So here's a voltage clamp recording to isolate the potassium current from individual cells. This is the total potassium current from a young cell, and in old mice, we saw there's a reduction in this fast, it's called KV4 component of the current. I mean, the overall current reduced, and then we isolate the TEA sensitive currents, and then the PETX1 sensitive current. There are different types of potassium current. They kind of all reduce with age. And this reduced potassium current will increase the neural excitability, basically makes the cell more excitable, and so that they can respond to even weaker input. So it's kind of a part of the central gain compensation to reduced sensory input from the cochlea. So that's what happens to the intrinsic properties of Bushy cells. And then again, overall, we evaluate the output of Bushy cells to a trance of auditory nerve stimulation. In young mice, we see that if you give a 100 Hertz trance stimulation, every stimulus, you trigger a nice looking action potential. It's very reliable, and it's temporally, it's very precise. This is 100 Hertz, and this is 400 Hertz. Still, it's very good. Even though there are some failures, we can see as the EPSC here, most stimuli triggered action potential in the postsynaptic Bushy cell. And in the old group, what we saw was that even under 100 Hertz, there are some failures. And at higher frequency of 400 Hertz, majority of the stimuli failed to trigger any action potential. And if you don't have action potential, that means the information stops at the Bushy cell. There's no signal passed to the upper nuclei, and information is lost at this sphere ganglia cell and Bushy cell connection. So what it suggests is that even though, I mean, even you have 100% input from the cochlea or from the auditory nerve, I mean, if this connection fails at the sphere ganglia cell, cochlear nucleus cell connection, and then you just lose the auditory information. And without information, you won't get the perception, and that's part of the hearing loss. So we think this morphological degeneration and physiological degradation of this sphere ganglia cell CN connection is underlying the development of age-related hearing loss. That in addition to the changes in the cochlea, I mean, at the hair cell and cochlear synapse level. Anyway, I want to summarize here that I showed you about the changes in the cochlea We know that there are damages in the inner hair cells and are conditions that even though the hair cells are fine, you can lose cochlear synapses, or you can lose spiral ganglion cells. And on the central side, this lost information will lead to corresponding changes at the central synapse terminal, as well as the connected postsynaptic neuron. They all show corresponding changes in terms of morphology. This is the synaptic degeneration. This is postsynaptic bushy cell degeneration. And also in terms of physiology. I didn't have time to show you what changes in synaptic transmission. It actually significantly compromised in aged mice. And postsynaptically, bushy cells show change the intrinsic property, and the readout of this whole system degrades over time in terms of firing rate and also the timing, the temporal information of this fired spikes also significantly compromised. So you're losing the rate, you're losing the timing, and then all this information will be reflected in compromised signal processing in upper nuclei. I mean, in the brain, ultimately in the auditory cortex. And just because you're losing information, you won't have good perception as young mice. And that's part of the age-related neuron loss. And our research just basically figured out in the cochlear nucleus what happens on the central side. And then it's kind of consistent with findings in the cochlea that in the past 10 to 15 years. So again, I want to thank everybody in my lab, especially two postdocs, Meijian Wang and Changeng Zhang. They did most of the work I talked about today. And here's my funding support. Yeah, thank you.